All right. Hello, we're live. And just to say to you out there in YouTube land, we're going to drop the exam Friday, and it'll be due Monday. And that's all I have to say about that. Look for outlines and stuff on Moodle. Thank you very much. All right. So, the bacteria, different bacteria require different kind of conditions. And it's one of the things that, there's so many different kinds of bacteria types that you can find bacteria pretty much anywhere on the planet in hugely variant environmental conditions. And B, they can be very specialized to their environment. Thinking about just on our, from pathogenic bacteria, because obviously we can talk about, geochemistry and bacteria in, underwater events and all this stuff. But pathogenic bacteria, our bodies are actually not particularly amenable places for our bacteria to live in. And just thinking about some of the bacterial or the environmental conditions, the skin is an interesting place to look, in terms of the oils and the pH and the all kinds of drynesses and salt and all kinds of things that make it so our bodies are set to limit some of the bacteria growth. That said, you are a walking massive bacterial contamination. In fact, if you want to think about it this way, really all you are is a way for your bacteria to get up and move around, because you are covered in bacteria. And one of the things I think is really very cool, just as a little side note, is I suspect when you are a little kid and you are home a lot, particularly little babies, there is probably a bacterial spectrum that you share with members of your family. And then as you get older, it might shift, but certainly as you leave the nest and you go off and you begin interacting with other people, you can slowly begin shifting your bacterial spectrum. You begin to smell differently. You have different bacteria on your body, or slightly different culture strains. And so it's not surprising that we might have really, local populations of bacteria that are specific to you and your family. And then that can shift as you bring others into your world. So, just a little thought there. As we're walking around, we're constantly dumping off bacteria and skin cells and dust and all kinds of stuff. All right, so lastly, when we think about what is required for growth, we have bacteria that require a very rich media, some do, in that they have a lot of needs. They're very needy little cusses, and you got to provide them with all kinds of stuff. There are other bacteria that are able to grow on really simple stuff. And everybody's going to need a source of nitrogen for proteins. You're going to need a source of carbon for all these other things. And then depending, there may be proteins that you can't make. There may be carbohydrates and carbohydrate types that are required. Different kinds of specific nutrient requirements. Carbon, hydrogen, oxygen, nitrogen, sulfur, phosphorus. Everybody's got to have that. And then we got these molecules that are found in smaller amounts. And micronutrients, very small amounts of things. If you're growing in the soil, these are all around. If you're growing in the seawater, these are all around you. We have very small amounts of these things as well. Sometimes you have fairly complex chemicals that are required, and that can make things really difficult to grow. So as we get further into the lab, we'll actually be using differential media and selective media. And this allows us to actually grow one specific bacteria over another. All right. So what we're now going to get into is this whole other thing, and that is metabolism. And bing. Actually, I'm going to do some of this on the board, some of this on the screen, mainly because I like writing on the board. And we'll start here. So this is a good chart to make up, and we're about to build this chart, in terms of thinking about metabolism. And it all comes down to thermodynamics. So we got anabolic metabolism and catabolic metabolism. And as I say, what we're going to do is, for my purposes, kill this, and I'll give you the PowerPoints, but taking notes is always a good thing. One thing with this kind of detail, being able to build both a list and we start talking about the metabolic pathways, being able to diagram out the metabolic pathway is the way you learn it. Just simply reading it and looking at it is not going to stick in your head very well. And what I hope is that as we do this, you'll see kind of the overall story, the theme of metabolism. And that's how you, I think, can best really begin to learn this. So if we start up here with metabolism as the reactions occurring in life, we have two different types of reactions. And just to throw back for a moment, it comes down to thermodynamics. And, oh, you can still see me on the board. All right. So the idea of thermodynamics has to do with what direction the reactions are going to go and whether they're going to require energy or they're going to give off energy. And so the first law of thermodynamics is not all that critical to what we're talking about, but the first law is stating that energy cannot be created or destroyed. So energy can be transferred to different forms, and that's all well and good. And it probably, it does play a role in how we're going to be thinking about our biochemistry and metabolism. But the second law is a really important one. And the second law states that all systems tend towards entropy, which is a fancy-ass way of saying disorganization. Now, what this means is we can think about reactions that are following the second law and being good little reactions, or reactions that are rebelling against the second law, bad little reactions. And if you're following the second law, that means, or one of the things we say when a reaction follows the second law is it is said to be a spontaneous reaction. That is kind of not a fair use of that term, spontaneous, because we tend to think of spontaneous as being something on the spur of the moment, something that happens immediately. In thermodynamics, spontaneous means it's not going to happen necessarily right away, but that's the direction it's going to go. Now, some things are really delicate, and they fall apart really fast. Consequently, you have to keep them frozen. And these are spontaneous reactions that are happening very quickly. But other things, they're just really tough. Now, they will break down eventually, like a chunk of wood, but it ain't going to break down fast. So, spontaneous reactions, then, are reactions that are occurring with this direction. And a spontaneous reaction, then, is going to be one that releases energy. And unless you're doing something fancy, like a cell, you generally release that energy as heat. But we can actually do work with it. So that's a way of thinking about this. Now, at the same time, a reaction that is non-spontaneous is a reaction that is actually increasing organization. It's like neatening up your room. A non-spontaneous reaction is going to require energy. So, we have these two different kinds of systems. One reactions that are giving up energy and reactions that are taking energy. And if we balance them, all is well. Now, under our grand chart of metabolism, we can begin to fill in some things like this. So we can lump all the biochemistry in living systems under this term metabolism, all well and good, but we've got two different kinds of metabolism that are important to distinguish between. We have a bad black marker. This is better. Better. We have what we call anabolic reactions, and then over here we've got catabolic reactions. You pretty much are going to be one or the other. Now, we can define anabolic reactions just on the screen there, we did, but I like to think of it this way, is we're going from simpler molecules to more complex molecules. So we're building stuff up. Catabolic is the exact opposite, breaking things down. So we're going from the more complex to simpler. All right. So in this manner then, if we're digesting stuff, breaking food apart, that is catabolic reactions. If we're building up all these biopolymers, that's anabolic reactions. Now, one of the things that we can then say, and this is a little way of thinking about this, when it comes to thinking about it and remembering which one's which, athletes might take anabolic steroids. I mean, if you ever hear about athletes taking steroids, they're taking anabolic steroids, and that's because they want to bulk up. Now, if you in the athletic biz, I mean, we shouldn't be taking steroids, but anabolic steroids do not turn you into the Hulk. What they do is they allow your body to recover more quickly, and they allow you to exercise at a higher, greater level and recover faster. And they probably also lead to cancer because of that. But regardless, that's what an anabolic steroid is doing. If you don't put the work in, just taking anabolic steroids is not going to do all that much for you. Catabolic, nobody takes catabolic steroids. Living is a catabolic steroid. Drinking, smoking, staying out too late, that's catabolic. So no, we don't do catabolic steroids. You do anabolic. Now, because this is violating the second law, we would say this is a non-spontaneous reaction. And because it's that, it is going to be requiring energy input. Now, over here, this is following the second law. This is a good little reaction. So this, we would say, is a spontaneous reaction. And these are going to be, then, of course, energy releasing reactions. Now, from both the bacteria's perspective and our perspective, if you balance these two things out, you're golden. If one side gets ahead of the other, at least for us, if you have more anabolic reactions, you start to get bigger. And if you are generating a lot of catabolic reactions, releasing a lot of energy, eating too much, you either are going to have that energy to burn or you begin to store it. So if we balance these two reactions, you stay right where you are in terms of like your weight, your body mass, and such. All right. So now, a couple other things fall out from this that are useful and worth throwing in here. These are energy requiring. These tend to be reduction reactions. And these tend to be oxidation reactions. And Tori's already heard this whole thing, but there you go. I don't know in chemistry if you've had this mnemonic or not, but this is my favorite, and it is oxidation is loss of electrons, and reduction is gain of electrons. And so we then use the mnemonic of oil rig. Yay! The other one that is sometimes used is loss of electrons is oxidation, Leo. Gain of electrons is reduction. And it's supposed to be Leo the lion says Gur, except that's not how you spell Gur, and so I can't abide by that one. But whatever works for you. So catabolic reactions are oxidation reactions. Anabolic reactions are reduction reactions. The only reason I say that is we're going to see, we start getting into metabolism, that there is both energy being generated and released, and there are electrons being harvested. And if you want to build up molecules, you're going to carry out reduction reactions. Reduction is a gain of electrons. You're going to need a source of electrons somewhere. And when you're oxidizing stuff, you're going to need a place to dump electrons, some place where you can actually dispose of the electrons. Electrons can't be just thrown into the air, exactly, or they don't just disappear. They're going to be put somewhere. So, next, we've talked a little bit about enzymes already in terms of the way they work. Yes. One of these things that at this point we're covering the exact same thing in molecular cell as we are in here, and I'm like, did I say this twice, or have I said it once to one group and not at all to you guys? We talk about the fact that enzymes can work as catalysts, and we'll throw this out just to make sure I have gone over it. Or did we say it here in this class or not? Under proteins.
Yeah.
That was in this class.
Mm-hmm.
Good. It was the same thing we said in cell though.
Yeah, on February 1st.
Basically, I'm the same thing over and over again. I mean, if you ask me, did you teach physics? Oh, sure. It'd be the same thing. Sorry. I'm a one-trick pony, man. All right. So, Tori says it. I believe it. We covered the fact that enzymes work because of protein structure. And what an enzyme is doing is lowering the energy of activation. What it means is we can carry out biochemical reactions at reasonable temperatures. Just as an example, if you want to break something apart, like molecules in glucose or in sugar, if you want to start breaking the carbons out, without enzymes you have to set it on fire, which is going to require 450 degrees. But with enzymes, we can actually carry it out at body temperature, or if you were to ask some bacteria out there, at this temperature, they might be doing a little bit, probably going very slowly, but certainly at like 40 degrees, they definitely are going. And this is how we can think about enzymes working, is that they're reducing the activation energy of a reaction, meaning that you lower the speed bump that you have to get over in order to have a reaction moving forward. Now, this is specifically for a catabolic reaction. Anabolic reactions are a little bit different, but for catabolic it works this way. All right, so enzymes can very often be phenomenally specific for individual molecules. In that, just as an example, you can take a stereoisomer, or any kind of isomer of like glucose, and that means it's 6 carbons, but it's rearranged in a different way. There are enzymes that can break down glucose really fast, but if you put in a stereoisomer of it, you can't touch it at all. It's like completely inert. So enzymes being very specific has a whole range of different consequences and stuff. Another thing, because this term a catalyst is what this is, it's not consumed by the reaction. So the enzymes are always present and hanging around. Now, we're going to get into a little bit of this later, but it's just a useful thing to think about. The fact that enzymes probably have to be regulated, and certainly they can be. Regulation is important because there are times when you need the enzyme around, and other times that you don't have the process going on. And this is sort of a nerd point, but it's interesting to think about. The production of proteins, the translation of RNA into a protein, is metabolically pretty damn expensive. It requires amino acids, it requires a fair amount of energy, and it is not something that a cell would do casually. And it's actually one of the cool differences between bacteria and more complex life. You and I, in our cells, we've got a whole range of proteins that at any given moment we're not using. But they are there, ready to be used in the case of an emergency. An adrenaline rush is a classic sort of thing. Or all kinds of different sorts of effects. You start to get really cold, or you start to get really hot. We got a variety of different proteins that are ready to go, but they're not doing anything. That is because we got enough metabolic energy coming in that we got a little bit of energy to spare. We can kind of operate this way. It's a very useful thing because, when the saber-toothed cat jumps out at you and you get this massive adrenaline rush, you want to be able to have enzymes that can break down storage molecules and dump glucose into your body within 10 seconds. As opposed to bacteria, they often have enzymes that, if they're not being used, they're not present. And so we have gene regulation, in which we actually use the gene to produce the enzyme, and we can regulate at the level of DNA, because if you don't need the enzyme, don't have it hanging around. And bacteria can get by with this because they are so darn fast at going from no enzyme at all to turning the gene on and producing a fair amount of the protein within 5 minutes. Whereas you and I, minimum 3 hours. I got my RNA-based vaccine on Sunday, and you could say, immediately, I'm feeling it. And, you're feeling the fact that you got jabbed with an inch needle, but there is no possible way with an RNA-based vaccine that you can have protein in under 3 hours. Just not going to happen. Because the processing and the events going on, no. And so by the end of the day, I'm going, okay, that's more than just the soreness from the jab. I'm actually starting to see the protein come up. Bacteria can get by with gene regulation a lot better than we can because they are just so darn fast at it. And that's not to say that we don't do this, because we got a ton of protein products that are not present in your cell until they get some kind of trigger, and then the gene gets turned on. And that's the difference between a fast-acting drug, like if you've ever seen or messed around with like asthma inhalers. You have fast-acting inhalers, which are opening up your bronchi really fast. Sudafed and adrenaline will do a brilliant job within seconds. And then you have slow-acting stuff. And slow-acting inhalers are often steroid-based, and they're slow-acting because they're turning new genes on. So there's a whole, some interesting stuff happening with that and some biochemistry. But if you've got an enzyme present, then you may only have it active at specific times. The enzyme can be inhibited. The enzyme can be activated. So you can have inhibitors or activators. And then we also have, what we call a feedback mechanism. It's a little bit like a thermostat. So if you think about the way a thermostat works, is you have a set point, and from a biochemistry perspective, you have a set number of molecules that you need to have floating around of this product. When the amount gets a little bit low, the enzyme cranks up and begins making more. When the amount reaches a set point, the enzyme shuts down. So it's not on all the time. And there's all kinds of different mechanisms by which we can regulate stuff. Okay, so that's just another way that we can think about some of these different reactions. Now, this is our overall goal. We're going to get into a range of different things. This is a couple of cartoons here of inhibitors. We can have a competitive inhibitor that is an inhibitor that will interact with an enzyme kind of like the authentic substrate, which, remember in our enzymatic terminology, that's the stuff that the enzyme is chewing on. So a competitive inhibitor would be like a sugar substitute. And this is not exactly right, but it's not bad. In fact, we actually use a sugar molecule in a lot of different bacteria gene expressions that the bacteria can't break apart. And a competitive inhibitor is one that if you add a whole bunch of the substrate, of the normal stuff, you can compete out the bad stuff. So in terms of poisons, I'll take a competitive inhibitor any day because a lot of different poisons end up being different kinds of enzyme inhibitors. If it's a competitive inhibitor, all you got to do is just feed the body a whole bunch more of the substrate. So like you put them on oxygen, or you hook them up to an IV with a lot of glucose, something like this. The other side of this is a non-competitive inhibitor, and a non-competitive inhibitor is going to essentially block the enzyme from working at all. It takes the enzyme out of action, and about all you can do there is wait it out. So these are different kinds of inhibitory systems, and we can have inhibition, but we can also have activation, things that will turn a protein on. And so this ends up developing kind of the possibility of very elaborate feedback loops in which you've got an enzyme that produces eventually a product. It's a part of a whole chain of things. And then this product is going to actually serve as an inhibitor of the enzyme, meaning that the thermostat kicks on, raises the temperature, and then the thermostat kicks off. Here, enzyme 1 kicks on, begins turning this process on to generate an amino acid. When that amino acid comes to a certain set point level, it is going to be acting as an inhibitor on the enzyme, and it shuts the whole thing down. So this allows us to have, or allows cells to have, a fairly fine control on the amount of things that are floating around. And then there's all kinds of, particularly in bacteria, of logic games where you can have activators, you can have repressors, you can have lots of different kinds of ways, little sort of essentially assembly line machines that allow these things to work in different ways. So we'll be getting into some of this. Now, we talked a little bit about this as well, in the fact that we use ATP as our energy system. And primarily, we use ATP because of the terminal phosphate. This phosphate bond is a high energy bond, and that's actually a pretty poor term. It actually breaks apart fairly fast. But it's the addition of that phosphate to other molecules, because the phosphate has what? Negative charge. Remember, we're supposed to always call out negative charge whenever we say phosphate. The negative charge can change protein shape. So, ATP is in fact a molecule that spontaneously decomposes really fast. We have ATP upstairs, but I keep it in the -70 freezer. I try not to freeze thaw it too often. If I need to use an ATP solution, I'll thaw it once, aliquot it out, split it up into a bunch of little tubes, put them back in the freezer, and take one at a time. Because the standard reason that your biochemical experiment doesn't work is your ATP is dead. So ATP, while it's got a high energy bond, whatever, really what's happening is that phosphate is transferring a tremendous amount of energy. And so these are some of the cast of characters in our grand tour of metabolism here. And let's see. There we go. This is what we're going to be getting into. We're going to simplify this. And we'll go back over a few things. But let's start out with some basics. All right. And then we'll get back into this. So, if you've had basic metabolism, then this is a review. If you haven't, well, this can be a little complicated. The level that we need to know for this specific class is not too high. We'll kind of break this thing down and talk about what goes in and what comes out at these different steps and see how that goes. So the primary set of biochemical pathways that we're going to get into for our work is going to be focusing on the breakdown of glucose. This is a pathway that is common to pretty much all organisms, short of viruses, on this here planet. So I always think about, if a bacteria and a giraffe were to walk into a bar, they don't have a whole lot they could talk about. Their lives are really, really different. But one thing they could talk about is the breakdown of glucose. Because pretty much no matter what kind of organism you are, breaking down glucose and breaking down other carbon molecules is something we all share in common. Everybody breaks down sugar. And the overall reaction is going to be actually similar to what you do to set fire to something. We're going to add, at least for our first part, oxygen. And then the reaction will result in carbon dioxide and water. And what comes out of this is a release of energy. Now, if you take a glucose polymer in the form of a piece of wood and get the activation energy up by using a match, you generate CO2 and water and you generate heat. So it's not anything that we have not seen before or have an understanding. What we're going to do, though, is capture this energy in the form of ATP, and we're also going to capture electrons. So, what we need to think about is the cast of characters in this grand play. And the opening character is going to be glucose. Just to throw this out, in case we haven't mentioned it, glucose is a 6-carbon sugar. And the only reason I mentioned the number is that we're going to want to watch because it's going to generate 6 carbon dioxides to see where they come from. All right. Now, another thing that we have to have in this grand scheme of metabolism, we're carrying out an oxidation reaction. We're going from a more complex molecule to a simpler molecule. And so that means that it's going to be releasing energy and it's going to be oxidation reactions. It's going to be releasing electrons. So if this is our opening star, another one of the members of the cast is a set of what we refer to as electron carriers. These are molecules that can pick up electrons and dump them off. Another way of saying this is they can be reduced, meaning they pick up an electron, and they can then be oxidized, meaning that they will lose an electron. And it is just one great big circle. They gain electrons, they dump them off, they go back and pick up some more. I tend to think of these guys as electron dump trucks, or electron garbage trucks. They go around scavenging up electrons. So we got electron carriers. The two main electron carriers that we're going to see in this game are abbreviated as NADH and FADH2. The last thing I'll just throw out here is their specific names. Just so if you see it, just say, oh, I know what that is. They are, actually, you'll see these as B vitamin derivatives. It is known as nicotinamide adenine dinucleotide. The structure is way more complex than we need to know. Let's just know that we refer to this as NAD when it's in the oxidized form. And when it picks up an electron, it's written as NADH. This is the reduced form. And it cycles back and forth between these two states. The other electron carrier, which you'll only see in just a couple of spots, but it is another B vitamin derivative called flavine adenine dinucleotide. And that, of course, is written as FAD. And FAD is how we write it in the oxidized state. And FADH2 is how we note it in the reduced state. So with that, we have a cast of characters that I think of as the grand musical theater play of metabolism. The last guy, which is sort of the, I guess in some ways the diva, and that is the energy-carrying molecule of adenosine triphosphate. ATP can then break apart to form adenosine diphosphate, and then you've got a phosphate molecule floating around. This is often abbreviated as a PI, which stands for inorganic phosphate, whatever. It's a phosphate molecule. And this can cycle back and forth as well. So when it does this, it gives off this phosphate, which is going to be able to drive different chemical reactions because it's a really big negative charge. And we can then reverse it back and remake an ATP molecule by adding it. So this biz, and one of the important things to think about is glucose is the one thing that's actually going in. Carbon dioxide and water are the things that are coming out. Everything else is just being recycled. So we've got electron acceptors that are cycling between the oxidized state and the reduced state and then back to the oxidized state. We have ATP that is cycling between the triphosphate and the diphosphate and the triphosphate. And you don't add extra stuff of those. Those are just set in the cell. The only thing we're going to add is glucose and oxygen. And the only thing that's going to come out is carbon dioxide and water. And this is a little interesting factoid for you and I. every single ATP molecule in your body is undergoing approximately 500 phosphorylation, dephosphorylations every day, or God, maybe every minute. Regardless, a lot. And it is said that we biosynthesize and rephosphorylate ATP to the tune of your body weight every day. Now, that doesn't mean that you're taking in fresh ATP, the equivalent of your body weight. That'd be really difficult to do. But we are constantly recycling it. So we're going to be getting into some metabolism on Friday and kind of put these things together into a grand musical and see where that goes. All right, so I'll catch up with you in a few hours. We're going to play with some microscopes and see what happens. And we'll be working with some dyes and stains. So don't worry anything that if you were to get a blue stain on, you'd be real sad about, because you might. That's all I know. Rock on.